In the last step of cellular respiration, we have what is called chemiosmosis. So this is the fifth step, and the theory of chemiosmosis was proposed by Peter Mitchell. So I'll write that right here. Peter Mitchell. And he won the Nobel Prize for this actually in 1978. So he said that the proton gradient provides the energy needed to phosphorylize ADP and synthesize ATP. So since the membrane in the mitochondria is impermeable to hydrogen protons because of cardiolipin. So cardiolipin is a weird lipid membrane that actually has four fatty acid tails instead of just two. So as you can imagine, the lipid bilayer is super tight, so you have all of these molecules here, and they're virtually impermeable, which is for a good reason in the mitochondria. So the pH difference that's created in the electron transport chain. So if I just draw this really quick right here, we pretend we have all of our complexes that I just talked about in the last video here. You have your four complexes here, and then let's pretend right here we have what is called the ATP synthase right here that I've just drawn. So you're creating this positive proton motive force here, right? So there's all these positive hydrogen protons here, and these are going to want to get back inside here, right? So how does this actually happen? Well, this is done through the ATP synthase here. So they provide the energy to synthesize large amounts of ATP. One mitochondria might contain 15,000 of these. So you can imagine how dense and how many there are of these inside our cells. So I'm going to talk briefly about the ATP synthase, which I will label up here. This is a very important molecule, and it's been studied by many scientists. So we have our ATP synthase. So you have your F1 domain. So this one has a bunch of different subunits. So this is a quaternary structure of proteins. If you've watched my protein structure, the different types from primary to quaternary, this would be a quaternary structure composed of different proteins that come together to form this big molecular machine. So there are 3 alpha subunits, so 1, 2, and then 1 in the back, so 3 right there. 3 beta, this is supposed to be beta here. My pen's acting a little weird, so that's beta, so there's 3 of those. There is a gamma, a delta, and then an epsilon, which I have neglected to draw because it is right here hiding. And put it in green. It is right down here. There's your epsilon right there. And then in your F naught domain, you have an A domain, C domain, which you have 10 to 14 of them, and then you have a B domain right here, which kind of acts like an anchor. So the gamma subunit right here is a long rod-like subunit that is like kind of shaped like a banana. So it goes from the tip of the F naught or the F0 all the way to the top of F1. The delta subunit is on the left side here, and it just helps with stability. And then epsilon is the one right here on the bottom right, helping anchor the F1 to the F naught. So I'm just going to call F0 F naught. The F naught domain, the A is the channel where the protons can move. There's 2 B's that reach up and hold on to the F1 domain, and then there's 10 to 14 C domains that are arranged in a ring, and they can all move around. So right here we are going to draw what actually happens to the ATP. I'll do it in blue. So these are the hydrogen, sorry. You have a hydrogen here. It is going to enter this little passageway here. So it comes in, and then is going to go in and bind to one of these domains. So all of these domains are going to be full with these hydrogen molecules. And each one of these little domains is called an asp61. So we will just draw that asp61. So there's this little opening door here that they go into, and then this is going to rotate, which I'll talk about in a second, how it actually rotates. And then there's going to be a little exiting door right here on the other side. an exiting door, and then this will bring it up here, and then your door out of the A subunit is here, and then this will leave, right? So it will leave right there out of the exit zone. So just to recap, the hydrogens move into the A unit, which is the entry channel. This does not pass all the way through A. It only goes about halfway, so it can't get up through here. The proton will veer off, and it will bind to one of the C subunits. When this happens, there will be a small conformational change. So it'll rotate about 30 degrees, right here. 30-degree rotation, and then this process will happen again and again and again. So there's a bunch of shifts until it'll eventually get to this exiting door, and this will exit. So the C units are spinning because the gamma right here, which is the F2 domain, the F1 domain, sorry, it will rotate. And as the gamma moves, since it is shaped like a banana, it will smash into the alpha and beta subunits, which are up here. Every time it bumps into one of them, it changes their conformation a bit. So there are a bunch of different conformations that can happen to the beta subunit, which is going to be our focus for this. The alpha subunits don't really do too much. The action kind of happens on the beta subunits. So they can take 3 different conformations, which I will draw right here. So we have this one, and then this one. I'll just draw the same thing. And then here. So it has 3 different conformations. The first one is called an open conformation. So when it is open, an ADP and a phosphate group can come into it. So you're going to have an ADP plus phosphate that'll come into it. The gamma will rotate, and the beta will change into another conformation that is called loose. So this is open, and this is loose. So this will shift to hold the ADP and the phosphate in place. So now they are stuck down here. They're stuck in this loose conformation to the beta subunit. The third and final conformation is called tight. Tight. And the ADP and the phosphate will be squished together, and then there's a spontaneous making of ATP. So finally, the gamma will hit again. The beta will go back into an open conformation, and the ATP will be free to go. So this will be in the open conformation. So each one of the betas is staggered. So if we have, I'll try and draw this here in 3 different colors up here actually. So we have one here that's like in that conformation, and then we have one connected to that right here. Oops. In another conformation, and then we have one right here in another conformation. So if this one's open, this one is tight, let's say, and this one is loose. So in this one, you won't have anything other than just a loose or a ADP or a phosphate sitting there. In this one, you're going to have an ADP and a phosphate that are held tightly, and in this one, you're going to have these ones are just anchored down in the ADP plus phosphate, and the tight is going to be anchored down, squished together to form the ATP, and then once it goes back to loose, it will be able to leave once again. So the way that they kind of proved that this ATP synthase rotated like this was called the actin filament experiment. So if you can imagine here, if I draw this little actin filament right here attached to the top of this F1 domain right there, you can see that they generally they modified an F1 portion of the ATP synthase. So they secured the actin filament to the top of the gamma subunits. They added ATP and found that the baby subunits did the same reaction, but they did it in reverse. So they added ATP. So the gamma piece rotated, and so did the actin filament, kind of like a propeller. So this was kind of spinning around and around and around, and they could see that with their probably electron microscope. So that is the end of cellular respiration. So you're going to make a bunch of ATPs from this step, which is the main ATP making step. So this is chemiosmosis.